This is Pennsylvania Inside Out on WPSU TV. Here's tonight's host, Patty Satalia.
Good evening. Coming up on Pennsylvania Inside Out, rebuilding the brain. Until recently, many scientists believed that the brain was unalterable. Once it was broken, it could not be fixed. But recent discoveries have convinced scientists to think differently. Evidence now shows that the brain remains plastic throughout life. That is, it can rewire or change itself in response to new learning. Under certain circumstances, the brain can even create new cells. What does this mean for those who have suffered brain trauma or for patients with Parkinson's or Alzheimer's disease? To find out more, we talked with Penn State's Gong Chen, an assistant professor of biology in Penn State's Eberly College of Science. A host of Penn State researchers are hard at work on unraveling the mysteries of brain disease, everything from why some people become addicted to alcohol to what causes anxiety and depression to why metals are important in normal brain function. Tell us a little bit about the focus of your brain research.
So we study molecular cellular mechanism of synapse formation and synaptic plasticity. So one of our research is how neuron forms connections, because that's the fundamental building block for the whole brain to function. So the synapse formation involves two neurons. One has so-called axon, another have dendrite. They contact each other. So a lot of molecular mechanisms still need to be unraveled. Why we study the synaptic functions, because in brain repair, when the new neurons born, they need to form connections with the adult neurons. So that's one part of our research. Another part, we study neural stem cells. So the neural stem cells have the capacity to generate new neurons, and the new neurons can form synapses with the adult neurons, so that to remake those connections and then regain the lost functions.
Before we find out more about exactly what you're doing in terms of synapses and plasticity and all of those things, you know, it's relatively easy, for example, to understand that the heart is a pump or that the lungs are bellows, but the heart, or rather the brain, which is really the master control center for the entire body, is more obscure, more veiled. We do know now that it is divided into regions, and we can actually map particular functions to particular sites, but overall, how great is our knowledge? How much do we understand about the inner workings of the brain?
Yes, we actually know a lot more about our brain than before. So, for example, the brain has billions of neurons, and each neuron may form thousands of synapses. So together, there are like trillions of synapses in our brain. And then we believe that when we learn new things and we memorize things, a lot of those information are stored in those synaptic connections. Like today I come here, this is my new experience. I believe like the hippocampus is one learning center. So probably when I see you and see these new settings, it's a lot of new things going on in my brain, particularly in my hippocampus. There are a lot of synaptic connections in my hippocampus get strengthened, whereas some others may be decreased. When I walk out of this room, there could have new synaptic connections being established, so that I can memorize today's things. And those are a lot of things really happening. Also like drug addiction. You would think that's addiction, but it's also a part of learning and memory. So when you have first experience, you love it, you get the drug again. It's kind of bad memory, as Dr. Kai Han said. We do know a lot about it. Also, the functional MRI can map our brain. What function, like, we fear something, those amygdala will light up. And then, if we have like anxiety, there are some other brain area, you will see more activity. So we really know a lot about it.
Well, until recently, though, scientists believed that brain cells were a finite resource. Unlike other parts of the body, those in the brain didn't regenerate. So in the event of a disease or an injury, once we killed those brain cells, that was that. Scientists now believe and know that we are regenerating brain cells. What's the implication of that? Why is that an important discovery?
Well, previously, the adult stem cells was not found, at least in the brain, was not found until 1960s, and then there are some signs of new neurons born, but scientists kind of like always doubt about it. That's the nature of scientists. We always keep healthy doubt, but until 1990s, the overwhelming evidence showing that there are new neurons generated, and they can survive a long time. They can even incorporate it. They form synaptic connections with adult neurons. Then people really believe it, and now the research is really thriving. A lot of studies revealing more and more those properties of the new neurons. And the new neurons are already being used to treat some the neurodegenerative diseases, such as the Parkinson's disease. A lot of studies already be going on.
In fact, you've said that thousands of neurons are added to the adult brain every day. This would mean then that the adult brain has some neurons that are days old, some that are years old.
Yes.
What's the difference perhaps in their function? Do they do different things?
Oh yes, those newly generated neurons are more plastic. So those newly generated neurons, they need to establish new connections with the adult cells. So they have a great capacity to form new connections, and those new synaptic connections store new information. So therefore, the adult neurons may already be saturated. They may already store some information there. So that's why I think kids can learn and memorize things a lot better than we adults. And when we get 80 years old, we probably cannot compete with the kids anymore, because they have much more capacity, the younger neurons.
So explain this plasticity. What makes them plastic, and what exactly does that mean?
So the plasticity has twofold. One is structural plasticity. Another is functional plasticity. The structural plasticity, as I said, is our adult neurons, although most of neurons are born in embryonic stage or even just immediately after birth. But there are constantly new neurons generated, and those new neurons form connections, and that synaptic whole neural circuit structure can be changed because of the new neuron add in. Another is functional plasticity. So although the synaptic connections form, it's not fixed. The synaptic connection can still be changed. The functional can increase or decrease. That's plasticity.
So for instance, if someone is blinded as a result of an accident, the part of the brain that would have controlled sight, now that the person has no vision, will that part of the brain likely take on another function?
Yes, actually, indeed the brain representation, each part of our body has the representation in the cortex. The blindness, our visual cortex would also corresponding to some other brain functions as well. In human being, probably it's difficult to study that, but we know a lot more about bird. We know that the Stanford researcher, they put a glass onto the owl, the night bird owl, put the glass there. The bird, the visual cortex and the audio cortex, they have overlapping functions. When they put a glass and then change the visual function, the audio function also change corresponding. In human beings, actually, we know more about this kind of accidental loss of arms. So both of our arms have corresponding or similar representation in our cortex. If there is accidental, one arm is lost. That arm corresponding representation area is shrinking, whereas another arm can expand to that territory. That is also another large layer of plasticity.
You're making me think of something else. We know now that the finite number of brain cells is not true, that they're always regenerating. But for a long time, we've always heard that we use only 10% of our brains. Is that true?
No, that's probably not true. I think, as far as I know, the reason for that kind of mythology is that when scientists doing some electrical recordings in the brain, they saw some neurons just quiet. Then at a given time, certain brain area may only have a small number of neurons firing. So they say, well, looks like a lot of neurons are quiet. No, that's not true, because each neuron have either particular type of function or they participate multiple tasks. At a given time, there is only a small group of neurons firing. But for example, if we see a green light, then one group of neurons will be activated. But when we see a red light, it's probably another group of neurons activated. You cannot say, when we see green, then the other part is not active, so they're not used. They're all used.
You mentioned a moment ago Parkinson's disease, and researchers I know have created Parkinson's-like disease in rat specimens, and then they used embryonic stem cells, and it appeared to have repaired the Parkinson's disease,
yes.
Although 10 weeks later they discovered that brain tumors began to grow.
Yeah.
So it sounds like some exciting things are happening, but proceed with caution.
Right. So stem cell research is still in its probably still infant stage, although it's really thriving. A lot of new studies, very exciting studies, found that the stem cells, if inject into the Parkinson's disease model, mostly in rats or mice, they can form dopamine neurons. That is what degenerated in Parkinson's disease. And those neurons do functionally integrate and then improve the symptom, like the rotation in the rat greatly reduced. So that's great. However, as any technology in the beginning stage, there are a lot of kind of pitfalls. Cancer is one thing, because stem cell and cancer cells share a lot of common properties. It's that they have this infinite
ability to do anything.
To renewal, right? So they keep generating. That could potentially lead to cancer. So scientists now put a lot of effort in how to limit that cancerous potential. One way is to induce them to differentiate. So once the stem cell differentiate into particular type of progenitors or precursor cells, they have the less ability to produce cancer.
So tell us a little bit about the practical or medical applications of what you're doing. You mentioned Parkinson's, but how else might the work you're doing now aid people with different brain diseases?
Yes, as we mentioned, I mentioned that we study synapse formation and synaptic plasticity. We also study stem cell research. What we are doing now is try to use the synapse formation, the knowledge we learned from our study between neuronal formation, then to apply it to stem cell research. One of our recent work, we can molecularly reconstruct synapses that normally formed between neurons. Now we can reconstruct in non-neuronal cells. That means we have learned a lot of those molecular building blocks for synapse formation. Now use that knowledge, apply to stem cell research. The stem cell, you cannot just inject stem cell, boom, you cure the disease. No, the stem cell need to differentiate into neurons. They need to survive. That's a big problem, because the new neurons, very difficult to integrate into adult network, because they have a good social there for a long time. Now you have newcomer. How you are going to integrate into that? That's a big problem. And that's we are studying how the new neurons form synapse with adult neurons. So that's our research. If we find that molecular mechanism, then we can inject the stem cells into, let's say, Alzheimer's disease model, and then try to help the new neurons to form connections with adult neurons.
Now, where do you think you're more likely to have a breakthrough? In something like Parkinson's disease or Alzheimer's, or a brain injury, something that was the result of a trauma?
Both Parkinson's disease and the acute trauma, like a stroke, they may have a high promise in the short term. Alzheimer's disease, a little bit more difficult, because Parkinson's disease involves one particular type of neurons, the dopamine neurons, in a particular area. That's from the substantia nigra projectile striatum. So we know that very well. It's focused, so once you know what type of neurons you want, and inject where, that's easy. Alzheimer's disease is more diffuse. We know cholinergic neurons is more important, but other neurons also involved. And more brain area may be involved. So that's a little bit more difficult. Stroke, on the other hand, we know it could be a small local area stroke, that's easier. But if it's, if it's large area stroke, that will be more difficult.
Now, we know that stem cell research is fairly restricted at this point. I'm wondering, at the pace that we're moving, how far out do you think it will be before there are really life-changing advances in medicine, for example?
I think there are a lot of promising studies. Almost every day there are new results coming. Unfortunately here, for human embryonic stem cells, there are ethical issues need to be dealt with, whereas other countries like Britain, they have more like liberal rules, so the federal money can be used for stem cell research. However, there are a lot of new studies like adult stem cells. That's, we can avoid those all ethical issues, and probably that adult stem cells has a higher promise, because if, like say, I have a disease, I use my own cells, that's probably most promising. However, it has a long road to go.
And what stem cells are you working with in your research lab?
We are currently using the rat adult stem cells that isolated from the rat hippocampus. In the adult brain, there are two main regions, subventricular zone and the hippocampus. The hippocampus is the learning center, and that actually the new neurons generated in the hippocampus is believed to related with the constant learning in the adult.
You're doing work with calcium phosphate uptake efficiency, which will mean nothing to someone who's not a scientist, but this has attracted attention around the world. Right. In layman's terms, why is that significant, what you're doing?
Okay, this is a kind of research tool that we can bring in the exogenous gene into a cell. So we do those transfection, like we want to know a particular gene function, we can either get another gene into it or mutate it. So this is a research tool. Previously, it's widely used in neuronal research because neurons are very difficult to get a gene in. But now with a lot of method we can do, but calcium phosphate is very easy, simple, and low toxicity. So we greatly improved the efficiency so that use this tool will be much easier to molecularly manipulate the cells, possibly including stem cells.
Now, fewer scientific fields are hotter than this stem cell research, but what kinds of additional research are needed, would you like to see being undertaken in places like Penn State?
Yes, the stem cell research is really very widely being pursued. A lot of states put a lot of money there. In fact, this year, Pennsylvania state also put in, I believe, quite some millions of dollars into the, the tobacco fund, into the stem cell research, cancer research. So Penn State, compared to many other like Harvard, Stanford, UCSF, we are still in the beginning stage, but there are a group of nice scientists studying stem cells. For me, neural stem cells, there are some other hepatopoietic stem cells and other regenerative medicine ongoing in Penn State, but we need a lot of more support.
So what's the take-home message? In what areas are you most hopeful, and what do you see as continued challenges as you move forward?
Yes, so I think, again, the most hopeful things are those very specific disease like Parkinson's disease. The clinical trials has been carried out in several hospitals, but the result is quite different, with some success, some other report failures. So it just tells you that although we are quite successful in rat Parkinson's model or mice model, but
not moving
applied
into
to human, there are still some practical problems. The Parkinson's still remain the most hopeful, and the stroke also very hopeful, because the stroke, The new finding is that in some brain, the stroke can trigger the endogenous neural stem cells, such as those in subventricular zone, to generate new neurons. And those new neurons can migrate to the injury site to do the self-repair. Now, the hope is that how can we stimulate the endogenous stem cell to generate more neurons, and then let the more neurons migrate to the injury site to do the self-repair. But unfortunately, those new neurons, most of them, 80% of them die. So we need to keep them survive and then let them form synaptic connections with adult neurons. Those are really hopeful. And Alzheimer's disease, we probably have relatively long way to go. But theoretically, I think every disease, they could have a potential to use the stem cell therapy.
All right. Thank you so much for talking with us.
Thank you.
A copy of the program you've just seen can be purchased through Penn State Media Sales at mediasales.psu.edu or by calling 800-770-2111.